Microbes glow in the deep sea, no magic, just chemistry. That seems impossible, yet it happens every night. How do bacteria produce light? The secret is a tiny enzyme system. Bioluminescence is the spontaneous emission of light by living organisms. In microbes, this glow results from a chemical reaction between luciferin and oxygen. The enzyme Luciferase catalyzes the oxidation of luciferin, producing excited state intermediates. When these intermediates return to ground state, photons are released. Microbes use this light for communication, predation avoidance, and symbiosis. Marine bacteria such as Vibrio fishieri illustrate how bioluminescence can be regulated by quorum sensing. Their luciferase genes evolved through horizontal gene transfer, showing adaptive benefits. Studying these systems reveals fundamental principles of enzyme, catalysis, and energy transduction. Because microbial genomes are compact, they provide a tractable model for genetic manipulation. The luciferase reaction is highly efficient, with quantum yields approaching 100% in optimal conditions. This efficiency makes it an attractive tool for bioimaging and biosensing applications. Thus, microbial bioluminescence serves both ecological functions and scientific inquiry. Pause 0.8d Luciferin, the substrate in firefly bioluminescence, is a bicyclic molecule. Its core consists of an indole ring fused to a thiazolidine scaffold. The indole nitrogen bears a carboxylate group that anchors the molecule. A sulfur atom bridges the indole and a carbonyl-bearing side chain. This arrangement creates an electron-rich system essential for light emission. In marine bacteria, the luciferase substrate is a fatty aldehyde. The aldehyde is produced from lucAB by the enzyme fatty acid synthase. Subsequent oxidation by an oxidoreductase generates the reactive aldehyde. Both pathways converge on a redox reaction, where luciferase catalyzes oxygen insertion. The resulting excited oxyluciferin emits photons as it relaxes to ground state. Key enzymes include luciferase, lucAB reductase, oxidoreductase, and fatty acid synthase. Understanding these biosynthetic steps clarifies how organisms tune their glow. Pause 0.8 Luciferases are enzymes that catalyze light emission by oxidizing luciferin. The firefly lucA family uses ATP and FMNH2 as cofactors to form a high-energy intermediate. In contrast, bacterial luxAB requires reduced flavin mononucleotide and a fatty acid substrate for the same oxidation step. Both enzymes share a Rossmann-like fold, but differ in key active site residues that stabilize distinct transition states. Firefly lucA contains a lysine that forms a transient phosphorybosylated intermediate, while luxAB uses a cysteine to bind the fatty acid chain. The catalytic pocket of lucA is lined with hydrophobic residues that accommodate the long-chain luciferin analogues found in insects. luxAB's pocket is more polar, allowing it to interact with the carboxylate group of its fatty acid substrate. Side-by-side overlays reveal a conserved glycine motif at positions FMN for electron transfer in both families. Despite these similarities, the reaction kinetics differ. Firefly lucA produces a flash, whereas luxAB generates a continuous glow. Understanding these mechanistic nuances informs bioengineering efforts to create custom, bioluminescent reporters. Pause 0.8 The catalytic cycle begins when luciferin enters the enzyme's active site. ATP binds to a secondary pocket, positioning the substrate for oxidation. An electron from luciferin is transferred to the enzyme's flavoprotein cofactor. Oxygen then reacts with the reduced cofactor, forming a high-energy peroxide intermediate. This peroxide cleaves the luciferin backbone, generating oxyluciferin and releasing energy. The excited oxyluciferin returns to its ground state, emitting a photon of visible light. Simultaneously, the enzyme is regenerated by reoxidation of the flavoprotein. The cycle repeats as new luciferin molecules bind and ATP is hydrolyzed. In bioluminescent organisms, this process occurs in specialized photocytes or organelles. The wavelength of emitted light depends on the luciferin structure and enzyme environment. Thus, the luciferase system couples chemical energy to photon production with high efficiency. Understanding this mechanism informs bioengineering of light-producing biosensors and imaging tools. Pause 0.8 In marine bacteria, Light production is tightly linked to cell density. The luxAPRIN encodes enzymes that convert luciferin into photons. Quorum-sensing molecules, called autoinducers, accumulate as the population grows. When a threshold concentration of autoinducer is reached, it binds to luxR. The luxR autoinducer complex activates transcription of the lux genes. This leads to increased synthesis of luciferase and the necessary substrates. Environmental cues such as temperature or pH can modulate this regulatory circuit. Higher temperatures often enhance enzyme activity, boosting luminescence intensity. Conversely, low pH may inhibit luciferase function and reduce light output. The resulting glow scales with cell density, forming a sigmoidal curve on the graph. Thus, quorum sensing ensures that bioluminescence is a communal signal. Pause 0.8 luciferase enzymes convert luciferin and ATP into visible photons, enabling quantitative readouts. In reporter assays, this photon output signals gene expression levels with high sensitivity. Biosensors use engineered luciferases to detect small molecules in real time. Microbial consortia can be programmed to emit light when metabolic pathways interact. Synthetic biology enables the assembly of orthogonal luciferase circuits within cells. These circuits translate environmental cues into quantifiable luminescent readouts. Future research seeks brighter, more stable luciferases for deep tissue imaging. Another goal is to couple light production with metabolic flux control. Such coupling could optimize biofuel synthesis by monitoring pathway activity. Ultimately, engineered bioluminescence may power autonomous microbial factories. By integrating light sensors into metabolic networks, cells can self-regulate enzyme expression. Additionally, multiplexed luciferase variants allow simultaneous tracking of multiple pathways. Pause 0.8's microbial bioluminescence relies on Luciferin-Luciferase chemistry, differing between fireflies and bacteria, regulated by quorum sensing, and used in diverse biotechnological tools.